So blood vessels are the delivery system of dynamic structures. They're dynamic structures that begin and end in the heart. See, the arteries, they're carrying blood away from the heart. And with the exception of the pulmonary system and the umbilical vessels in the fetus, they're going to be carrying oxygenated blood. Capillaries are going to be the direct contact to the tissue cells, and they're going to be directly serving cellular needs by delivering oxygen to those tissues and removing carbon dioxide wastes. Oxygen is going to be the main delivery of these capillaries. However, glucose, amino acids, and various other needs are going to be served by these cells, so by these tissues. Now, veins are going to be carrying blood toward the heart, and their main job in the tissues is to carry away carbon dioxide wastes, as well as nitrogen wastes and amino acid byproducts. So this is going to be a broad strokes overview of the systemic circulation. This is just the systemic circulation. We have removed in this image, the pulmonic circulation. So we have our heart, our left heart, and our arterial system is going to start with our elastic conducting arteries, our elastic arteries, and they're going to move to our muscular distributing arteries. And then from there, we'll have some arteriovenous anastomoses, which there will be arteriovenous anastomoses periodically throughout the entirety of the cardiovascular system, but we don't get too deep into those in AS Bio, so I'm not going to get too deep into those right now. But suffice it to say that an anastomosis is just a direct connection between an artery and a vein with no capillary bed in between. So you are directly moving arterial blood to venous blood without any gas exchange occurring in between. Now, this break right here is just showing distance. It's just a way of showing distance. Now, from this show of distance, we're going to be in completely we're just going to break down and get smaller and smaller and smaller until we reach our resistance vessels, our arterioles. Our terminal arterioles are the last arterioles that we're going to reach until before we reach capillary bed. A met arteriole is going to be the arteriole that will branch into our precapillary sphincters. Our precapillary sphincters are going to branch into our true capillary beds. And these will have these smooth muscle sphincters, which are just rings of smooth muscle. And when they pinch off, they will close off the true capillary bed. Our metarteriole is going to continue and become a thoroughfare channel, which, if we close off our precapillary sphincters, that will become effectively a shunt of metarteriole to thoroughfare channel, and that will directly flow into our postcapillary venule. Now, these sinusoids that we see here, those are just expansions. Those are just little bulges in the capillaries that we'll see sometimes. So a postcapillary venule is the venule that will receive the blood after a capillary. That will flow into a vein, the small capacitance vessels that will receive that blood. Those small veins will lead to larger and yet larger and yet larger veins until we reach to our venous system that drains directly into the heart, this being our right heart. Now, here we have our lymphatic system. Our lymphatic system, lymph is Latin for water, and our lymphatic system is a series of tubules, blind-ended ducts, that draw away excess fluids from the tissue beds. And those excess fluids will be drawn along a series of nodes, fleshy nodes, that have an immune behavior associated with them. And these fleshy nodes we'll get more into them in Unit 5, but these fleshy nodes will eventually lead one to another, to another, to another, eventually crisscrossing to a point, and they'll all get to, they'll all drain eventually into the thoracic duct and then into the heart. Not all of them will drain into the thoracic duct, to be fair, but the largest one is the thoracic duct. And they'll all drain back into the heart. So all the lymph will eventually flow back into the heart. So that effectively all of the excess fluid is going to drain back to your circulation. You really don't want to have excess fluid floating around, because too much fluid floating around causes problems. Now, let's go more in detail about the structure of those blood vessel walls. I mentioned before, just a few moments before, about the tunica intima, media, and externa. Now, the lumen, as I discussed and I sort of alluded to, is that central blood-containing space. And those capillaries are endothelium with a sparse basal lamina. So this is a more detailed image of what I showed you before. In the tunica intima, we'll have an endothelium. This is a single layer of endothelial cells, much like what we'll be seeing in our capillaries. This is continuous with our capillaries, in fact. In large diameter, 1 millimeter diameter or greater vessels, we will have a subendothelial layer. Now, that will not get you points on the Cambridge exam. That is just nice to know. You'll have a subendothelial layer in large arteries and veins. Also, in large arteries, you'll have an internal elastic lamina. By large arteries, I'm referring to the elastic arteries, the conducting arteries. All right? You'll have an internal elastic lamina. Then you will have your tunica media, which is smooth muscle and elastic fibers. Then you're going to have your external elastic lamina. This is still only in conducting arteries, the elastic ones. In a muscular artery, the internal and external elastic lamina are going to not be there. They're going to be absent. You are still going to have your tunica media. In an arterial, you're not going to have the internal and external elastic lamina. You will still have your tunica media. These two are going to be absent in everything but the elastic arteries. Now, the tunica externa is present on the outermost layer. That is your tunica externa. It is comprised primarily of collagen fibers. Now, same thing goes for your vein. You'll have your tunica intima with your endothelial layer, your subendothelial layer in large vessels. Your tunica media will be present. There will be no elastic lamina in any vein. And your tunica externa will be present, comprised of the same stuff. Your capillaries will always be comprised of endothelial cells, a single layer, and a thin basement membrane. Now, the endothelium will line the lumen of all the vessels, this tunica intima. And like I said before, in vessels larger than 1 millimeter, a subendothelial connective tissue basement membrane is present. The tunica media is a smooth muscle with sheets of elastin. It is responsible for vasoconstriction and vasodilation of the vessels. The tunica externa, and as I often call it, the tunica adventitia, those terms are interchangeable, but on the exam, please only stick to one. If you're going to choose a term, as I've said in a previous video on this channel, stick to one term. If you're going to be American, be American. If you're going to be British, be British. If you're going to choose a term, choose a term. Pick a side. Die on that hill. All right. So, collagen fibers are present to protect and reinforce that vessel. In larger vessels, you will have an interesting adaptation to nourish the external layer. In very large vessels, you'll have something called the vasa vasorum. Vessels of the vessels. Vasa vasorum are vessels that are existing solely to feed the large vessels. There are branches of these arteries and veins that exist to feed the vessels themselves. The vessels are so large that they need their own blood supply in order to survive. The thickness of the artery is such that diffusion alone is not enough for it to get the oxygen it requires for the tissue on the outside of the artery to stay alive. So vasa vasorum are required. Now, this is a summary of the makeup of certain blood vessels. Elastic arteries have more elastic tissue and less smooth muscle than muscular arteries by comparison. You can see that the ratio shifts significantly. They also have less fibrous tissue than muscular arteries. So that makes muscular arteries thicker and stiffer than our elastic arteries. These elastic arteries are more rubbery and have more of a recoil force, whereas muscular arteries are able to generate more pressure internally and are able to have a smaller lumen diameter. Arterials have a similar makeup to muscular arteries, but they have less smooth muscle and less collagen overall. Now, in capillaries, there is no elastic tissue, smooth muscle, or collagen because, as we discussed, they are all endothelium all the time. In venules, we have endothelium and smooth muscles and fibrous tissue, but we do not have elastic tissue. We have some elastic tissue in the tunica media of large veins, but we mostly have smooth muscle and fibrous collagen. We have more collagen than anything else because it is not the job of veins to provide muscular contraction and therefore pressure. It is the job of veins to allow for blood to flow at low pressure into the veins. Now, let's talk about the arteries themselves. Let's talk about elastic arteries first. We have large, thick-walled arteries with elastin in all three tunics. The aorta and its major branches are our elastic arteries. It has a large lumen compared to other arteries. It acts as a pressure reservoir. It expands and recoils as the blood is ejected from the heart. We have muscular arteries and arterioles. They are distal to the elastic arteries, and by the way, proximal means closer to, and distal means farther from. Proximate, proximal, approximate, close to. Distal, distance, farther from. So, distal to elastic arteries, they will deliver blood to body organs. They will have a thick tunica media with more smooth muscle. They are very active in vasoconstriction. They will constrict the vessel. Now, arterioles, these are the smallest arteries, and they will lead to your capillary beds. They control the flow into those capillary beds by means of vasodilation and vasoconstriction. Capillaries themselves, these are the microscopic blood vessels. They have walls of thin tunica intima that are 1 cell thick. Sometimes they are so small that they will only allow a single red blood cell through at one time. Every tissue in the body will have capillaries, with a few exceptions. Cartilage does not have capillaries. Epithelial tissues does not have capillaries. The cornea and lens of the eye do not have capillaries. Now, the cornea and lens of the eye makes sense. Otherwise, you would basically have vessels constantly in your vision. It would like be trying to see through a bramble patch. That would sort of be in the way. Oh no, look, a lion. Yeah, pretty much. So, the function of the capillaries is to exchange gases and nutrients and wastes and hormones and all that other stuff. Isn't that nice? That's nice. I think so. Now, see this again? I think we've pretty much come all the way half circle at this point. Yeah, half circle. We're at the capillary beds still though. So the capillary beds, they're interwoven networks of capillaries. They form the microcirculation between the arterioles and the venules. We have two types of vessels in this world. We've got the vascular shunt, which is the medarteriole and thoroughfare channel, and this is directly connecting our terminal arteriole to the post-capillary venule. And then we have our true capillaries. Every capillary bed has about 10 to 100 true capillaries. These are our exchange vessels. They're going to branch off of our medarterial or our terminal arteriole. So, as I mentioned before, we have our terminal arteriole here, and we have our post-capillary venule here. We have our precapillary sphincters. If our precapillary sphincters constrict, that will cause blood to stop flowing from high pressure to low pressure in the capillaries. And then all of the high pressure and the low pressure will go from arterial to venule. And no pressure can go into the capillaries. So no blood will flow into the capillaries. But why won't blood flow backwards? Well, here's the problem. Let's say, for a moment, that our pressure in our arterial is 1, and our post-capillary venule is -1, and our terminal and our capillary pressure is 0. You can't have pressure flow from -1 to 0. That's not how it works. You can't flow passively from low to medium, back to high. You have only from high to low, down a pressure potential gradient. So when you block off a path of least resistance, you will always flow through the path that is now the path of least resistance. This blood is going to flow through the path of least resistance. And that path of least resistance is going to ignore these side paths that are now higher pressure than this direction. So you only need to block off the terminal arterial end. Now, venules are formed when the capillary beds unite. Venules are porous. They allow fluids and white blood cells into the tissues. The post-capillary venules are consisting of endothelium and a few pericytes. Larger venules have 1 or 2 layers of smooth muscle. So veins are formed when venules are coming together. They'll have thinner walls, larger lumens compared to their corresponding arteries, and the blood pressure is lower than in arteries. So this is once again our thick-walled artery, our thinner-walled vein, our larger lumen, our smaller lumen. Now where is all the blood? If all of our lumens in our veins are larger, well, suddenly it makes sense that 60% of all of our blood is in our veins. Our systemic arteries and arterioles only have 15% of our blood. All of our pulmonary blood vessels only contain 12% of our blood because I don't know if you've measured your chest recently, but it doesn't really have all that much, it doesn't really take up all that much space on your entire body. Most of you is not your lungs. Your heart contains 8% of your blood at any given time. Now that's a significant amount of blood considering your heart is only about the size of a closed fist. Your capillaries contain about 5% of your blood. That ain't much. Your veins are where most of your blood is. Now, veins will have adaptations that make sure the blood goes back to the heart. They have a large diameter of lumen, so they offer very low resistance. And they have valves that prevent the flow of blood backwards. They're mostly abundant in the veins of the limbs, your arms and your legs. Varicose veins will develop when the valves of your limbs fail. So this is the internet. I'm sure you can find pictures of varicose veins. But please, safe search on. Now, let's talk about some physiology of circulation. The definition first. Blood pressure. Blood pressure is a force per unit area exerted on the wall of a blood vessel by blood. It is expressed in millimeters of mercury, measured as a systemic arterial blood pressure in large arteries near the heart. The pressure gradient provides the driving force that keeps blood moving from an area of high pressure to low pressure. So blood pressure is what moves your blood through your heart, recycling all the blood in your body about once every minute. Now, this is how I used to have to take blood pressure back in the 1700s, because yes, I pretend to be that old. Now, I'm really only 251. I'm not 200. I'm not 270. I'm lying. All right. Now, the Reverend Stephen Hales invented the first technique to measure blood pressure in the 18th century. His technique involved placing a glass tube on in a horse's carotid artery. This was a one-off measurement